Now, the triploid primary endosperm nucleus divides quickly into the thousands of free nuclei. Did you observe one thing over here, students, that we are not talking about cell division. I'm talking about nucleus division, because what has formed? It was a primary endosperm nucleus that has formed. This primary endosperm nucleus keep on dividing and they form a large number of nucleuses present in that embryo cell. So basically, without having a complete cell formation, there is only the nucleus that is continuously dividing and we are getting a large number of free nucleus. Remember that they are free nucleus without any covering of the cell wall. Here we get confused at how this is dividing. They were triploid in nature, they keep on dividing and they quickly forms thousands of free nucleuses in the embryo cell. Now, all these nuclei remain in the liquid form, or which is called as acellular form. There won't be any cell development. In later stages, cell wall formation may take place. So initially, what happens when the 3N nucleus is dividing, when the 3N nucleus is dividing, let us consider this, they keep on dividing. Multiple times they divide, and what they form? They form thousands of free nucleuses. What are they? These are nothing but nucleus. They are not the cells. They are not the cells. Okay. And this is the most common type in the endosperm formation. So the later stages, in some cases cell wall formation will take place, but initially it was only the free nucleus which will be present. And in which form they will be? They will be in the liquid form, or they will be in the acellular form. There won't be any cell development. Fine. Did you understand this? What is happening in endosperm formation? The endosperm is present in the liquid form. They are not forming the cell immediately. Okay, let us see what we have next. Now, the number of free nuclei formed varies depending on the species. What type of nucleuses they are going to form? That depends on the varying species. One species endosperm formation with the other species endosperm formation, if you compare, you will find a lot of difference over there. It is not always same. The best example what we can give over here is about coconut. The liquid, whatever we get it in the form of coconut water, that is nothing but the endosperm present in the, where it has divided multiple times and it is in the acellular form. We say it is filled with nutrients. Yes, it is filled with nutrients because it is the primary endosperm nucleus that has divided and we are actually having the acellular form that is in the free nuclear form. As division progresses, the nuclear push towards the periphery and the center will be occupied by a large vacuole. So what happens when the division is continuing, when the endosperm formation is continuing, the nucleuses are slowly pushed towards the periphery of the embryo sac. So if at all there is an embryo sac, now no cells are there, no antipodal and no synergids because they have already degenerated. Their work is done. Now what is there? There is only free endosperm nucleus, or the thousands of free nucleus is present, and with that there is a zygote which is 2n in number. This is present. Okay. So now what happens after that? This nucleuses which has divided and made thousands of the copies, they will be pushed towards the periphery. Pushed towards the periphery means they will go to the last part or the peripheral part of it, making a large vacuole inside. What they are going to make? They are going to be making a large vacuole that will be occupied by a large vacuole. Clear? What is happening? The endosperm formation is taking place. From where the endosperm formation is taking place, what we got it in the form of a primary endosperm nucleus where ploidy was 3N, that is combination of a two polar nuclei and a male gamete. We got a 3N ploidy containing a nucleus, and that keep on dividing to form this endosperm. Clear till here? Don't get confused. These concepts are very, very important, and this will be there for, like, you know, it is so important that on this you may get questions again and again repeatedly asked in different exams. Okay. Now, cell wall formation takes place from the periphery to the center gradually, making the entire endosperm cellular. Now, as it was pushed towards the periphery, what has happened? The cells are already gone to the peripheral side, right? The nucleuses has been pushed towards the periphery. Okay, these are the nucleuses which has been pushed towards the periphery. Now slowly there will be a gradually formation, cell wall formation will take place. On this there will be a gradual cell wall formation which will take place now. Okay, so if I wanted to draw the cell wall, so on this there will be a gradual cell wall formation. How it will start? It will start from the periphery and it will go towards the center, and slowly it may cover the entire endosperm. Complete endosperm, slowly it will be getting covered by the cells. Now the cells will start forming. So what as initially was that? Initially it was only the nucleuses. Nucleuses has thousands in number. They are in acellular form. They have been pushed towards the periphery, and once the stage has come, then slowly the cell wall formation will start, and it will start from the from where it is on the from the outer side, that means from the periphery towards the center. Slowly it will come and occupy the whole endosperm. The central in coconut, the outer white kernel is the multicellular endosperm. What we eat it in the coconut, when the coconut is completely ripened and we get the white color kernel, right? That is nothing but the endosperm. What is it? They are nothing but the outer thing. That is a multicellular endosperm that is eatable, full with nutrients. Okay. The central liquid in the coconut water is the free nuclear endosperm made up of thousands of free nuclei. I told you, right? In the center, whatever we get it in the liquid form is nothing but the free endosperm nucleus. It is in a acellular form. It is in a liquid form. That is also containing a lot of nutrients. Did you understand? So what has happened now? Let us quickly one more time revise. Primary endosperm nucleus has formed, that started dividing thousands of times. We have got lot number of nucleuses without the cell wall. After some time, that will be pushed towards the periphery. In between, we will get a large vacuole. Slowly now the cell wall formation will start, and it will continuously divide, forming and covering the entire endosperm. Okay. In case of coconut, we see that thick multicellular endosperm formation in the form of white kernel, the eatable form of the coconut. And the central liquid of the coconut water is nuclear endosperm, which is made up of thousands of free nuclei. Okay. So I hope you got it till here. Let us see what we have in the next slide. Okay, now the central nuclei has formed, right? Now the central nuclei is, the central part is filled with the liquid, and that liquid is nothing but the free nucleuses. They don't have any cell wall formed over there. So in coconut, this is a specific capacity what we see in case of endosperms. Okay, so this is the matured embryo which is shown, where we have the endocarp, embryo, exocarp, mesocarp. I'll tell you about it later, because now right now, just for your in a reference, I'm keeping the picture. Now, post-fertilization events are already been started, right? When it was started? Post-fertilization events started after the double fertilization itself. As soon as the double fertilization and triple fusion finishes, we start with our post-fertilization events. Okay, so we are almost at the last part of the fertilization events, all the fertilization events. We have discussed pre-fertilization events. We discussed about fertilization events and how does the fertilization take place. And now we are at the post-fertilization event. The cells of the endosperm store large quantity of nutrients. I told you already. The quantity and the nature of the endosperm may vary depending on the plant. This also we have discussed. Now, mostly the endosperm is used up by the growing embryo, and it is no longer seen in the mature seed. This endosperm is meant to help the embryonal growth. So most of the plants uses up this whole endosperm completely while developing the embryo. Okay. But in some plants like ricinus, it forms a permanent storage tissue called as perisperm, which persists till the germination of the seed. Okay. Sometimes they give this, what is perisperm? Okay. Sometimes they ask this. Okay. What is perisperm? This is done. Okay. Now, what happens, in some cases, like in the specifically in the plant like ricinus, the endosperm will be there at the end of the completion of the whole process also. That will make a permanent storage. That perisperm will make the permanent storage, and this will persist till the germination of the seed. So what is perisperm? Perisperm is the leftover endosperm which is not utilized by the growing embryo. I'll repeat again. What is perisperm? Perisperm is the part of the embryo, a part of the endosperm nucleus or part of the endosperm which is not utilized till the germination of the seed, and that is called as perisperm. Where do we see this? We see this in the case of ricinus. Ricinus is the plant where we see that endosperm is still persisting or still present over there. Fine. Is it clear till here? Now, the cells of the endosperm store large quantity of nutrients. Well, this slide is coming again and again. Okay, the which persists till germination. This is also done. Let us see the next one. Okay, now we have the embryo formation. We learned what we have to learn now in post-fertilization event. We have to talk about endosperm formation, embryo formation, seed, and the fruit formation. Fine. So we have 4 more events to go, like 3 more events to go. One is done. Let us see that the next one is embryo formation. Perisperm is required for the growth of the developing embryo. Embryo will wait till the perisperm is mature. Perisperm is completely required. After that, the embryo formation will take place. The process of development of mature embryo from the zygote is called as embryogeny. Very important definition. What is embryogeny? Embryogeny is the formation of the embryo. Formation of the mature embryo is called as embryogeny. The process of development of mature embryo is called as embryogeny. Fine. This is from the zygote to the embryo formation. That is embryogeny. After the syngamy, that is a double fertilization, the zygote undergoes a period of rest before it starts dividing. I told you the zygote will not start dividing immediately. It will be waiting for some point of time so that it gets a complete endosperm development. Okay. Next, what happens? What happens if it after taking the rest? After the resting period is over for the zygote and endosperm has completely prepared, the zygote varies in different species. The resting period of the zygote will be different in different species because see, the endosperm formation is also, time is also different. How much time does it take to form an endosperm is different from the time that the zygote is going to be formed. So that is why resting period for a zygote will be different from different species. Okay. And that also depends on the environmental condition. If the conditions are harsh or if the plant is not having proper food and all, the development period also will prolong. The zygote to embryo development period may become more or the resting period may become more. Okay. Clear till here? In general, primary endosperm nucleus divides first and the zygote divides shortly afterwards. Generally, what happens once the primary endosperm nucleus starts forming, just immediately after that, the zygote also will start dividing. Okay, so that complete process keep on happening. So first, the primary endosperm nucleus will be dividing, and followed by that, the zygote will also start developing. Okay. The first division of the zygote is transverse and resulting in 2 cells. Okay, from here we have to start understanding it very properly, how the embryo formation is taking place. Now there was the zygote. Okay, inside the embryo sac there was a zygote. This is 2n in number. Fine. This zygote now will follow a transverse division, not a longitudinal but a transverse division. Like it is longitudinal and transverse, it will go a transverse section. So this embryo will start dividing in a transverse section like this. The zygote will give a division of a transverse section. In between there will be a division. Okay, and that will be forming 2 cells after that. One is called as embryonal cell, the other one is called as suspensor cell. What it is? One is called as embryonal cell, another one is called as suspensor cell. The cell which is towards the interior of the embryo sac is called as embryo cell or the terminal cell, and the other cell which is towards the micropylar end is called as basal cell or the suspensor cell. Very important thing, you should understand this concept very clearly. Okay. Let me go to that slide and I'll tell you how this process is going on. Okay. Where we are actually in this slide. Okay. The embryo formation. Okay. This is done. This one. Yeah. Now in this cell, now the cell which is towards the interior of the embryo. Here the embryonal cell started dividing. Can you see? Now this was the embryo. It has gone a transverse division. Okay. Now which is towards the embryo sac is called as embryonal cell, which is towards the embryo sac is called as embryonal cell. If I think like this, this is the embryonal cell. Okay, this is the embryonal cell, and the down one, the cell is called as that is called as basal or basal means it is present at the base, right? So it is called as basal cell, or it is also called as suspensor cell. Okay, it is called as basal cell or the suspensor cell. So I am talking about this A diagram only over here, the first diagram. What is happening? The zygote now started dividing. That will be a first transverse division. There will be a first transverse division that will take place,